continue with the cell division, where there are two processes through which cell division happens in human body or plant body or animal body. So, it is mitosis and meiosis. During this first series, we will be more on mitosis and then we will continue on meiosis, and why there are two different processes we will discuss in detail. So, mitosis, it is a equational division. The term was given by Fleming. Mitos, a Greek word, means thread or fibril. So, it was studied by Strasberg in plant cells and Boveri and Fleming in animal cells. So, what is mitosis? It is a type of division in which chromosomes replicate, become equally distributed both quantitatively and qualitatively in two daughter nuclei, so that the daughter cells come to have the same number and type of chromosomes as present in the parent cells. There is no change from the parent cells to the daughter cells. So, that is why it is called equational division. The number of chromosomes remains the same. The type of chromosomes that has been transferred to the daughter cells would exactly be the same as the parent cells. So, the distribution is both qualitatively and quantitatively equal between the two daughter cells. So, mitosis occur in the formation of somatic body cells. So, it is also known as somatic cell division. So, the sites of mitotic cell division in plants are basically, which are growth regions, meristematic regions like stem tip, root tip, intercalary meristem, lateral meristem, growth of embryo, leaves, flowers, fruits, seeds, or etc. So, basically if we try to understand that mitotic cells happens where there is a growth or a growth kind of thing is required, mitotic cell division happens at those places. The sites of mitotic cell division in animals are diploid cells like embryo development, some restricted regions in the mature form like skin and bone marrow. In haploid cells, somatic cell division is rare, but one of the few examples is male honeybee or drone production. Mitosis take 30 minutes to 3 hours for completion depending on the type of cells and the type of species. During mitosis, plant cells does not show much change, while animal cells become spheroid, more viscous and reflectile. So, in mitosis there are two further phases, karyokinesis and cytokinesis. Cytokinesis is the division of cytoplasm, karyokinesis is the division of nucleus. Karyokinesis, indirect nuclear division, karyon again a Greek word which means nucleus, kinesis means movement. So, the nucleus passes through a complicated sequence of events before forming two daughter nuclei, which are prophase, metaphase, anaphase and telophase. Sometimes there is pro-metaphase also. These are actually not a phases which have gap in between or which can stop after another phase, like prophase. Once the cell has entered the prophase, then metaphase can happen or not happen, that is not a question, it will happen. These phases are just named to understand easily what karyokinesis is all about. These are all interconnected phases and there is no gap in between them. So, this is a basic understanding or basic overview of all those phases. We will discuss these phases one by one, but if you can see from these slides, there are changes in the nuclear membrane, nucleolus, spindle formation, astral stars, movement of chromosomes, and then the generation of cell membrane or the final cleavage that is being happening. So, this is an overview of mitosis happening in an animal cell. So, there is some difference of mitosis in plant cells, we will discuss in the coming slides. So, the prophase, as the name suggests, pro means first and phases is stage. So, it is the first stage of mitosis. Viscosity of cytoplasm increases, In the initial stages, we will discuss everything about the animal cell division, mitosis happening in animal cells. So, viscosity of cytoplasm increases when it enters into the prophase of cell division. Indistinct and intertwined DNA molecules condense to form elongated chromosomes. And further in prophase, the chromatin reticulum disappears and the shortening and thickening of chromosomes occur. And why this occurs? It becomes because coming together of scaffolding or axial proteins. So, the role of proteins come here, which are causing the shortening and thickening of the chromosomes, twisting and coming together of lateral loops of chromatin to form the filaments. So, at this stage and post this stage, you can actually differentiate between different chromosomes. You can actually look into the different chromosomes, because before this stage the DNA has been coiled itself and they are indistinct, and you actually can't differentiate between different chromosomes, but reading the chromosomes post this phase becomes little easier. And this thickening or condensation of chromosomes is assisted by proteins condensins. In the beginning of prophase, animal cells have two centromere or centriole pairs close together. So, what we have seen in G2 phase, the centrioles have started preparing itself for the M phase. So, in prophase, we can see that there are two centromeres or centriole pairs which are close together. So, now they have started, after the prophase, they will start separating from each other. The two begin to shift towards the opposite sides, and both the centriole pairs radiate out fine microtubule fibrils called estral rays. So, there is a difference that in plant cell division, we will see some changes in the prophase, that what makes it different from animal cell division. So, these microtubular fibrils are playing a very important role in cell division. They separate out the two divided chromosomes into two different daughter cells by joining through centrioles. So, each group of estral rays along with its centriole pair is called an ester. In an ester, the microtubular estral rays are not connected to centriole but to the pericentriolar satellites. And what all happens in the prophase is that chromosomes are evenly distributed inside the nucleus, whereas in middle prophase they shift towards the periphery of or the nuclear envelope, so it leave a clear central area. So, there is nothing in the central area of the nucleus. They start moving towards the periphery of the nuclear envelope, and it is called the middle prophase. And the chromosomes shorten and thicken further to assume characteristic shape and size. So, what their shape is and what their size is, we can clearly we can see them. And the size of the chromosomes, if we try to really understand them in number, the size of chromosome is reduced to 1/25th of their size in early prophase. So, they shorten so much that the size is reduced to 1/25th. And each chromosome appears to consist of two longitudinal threads called chromatids. So, these are the two chromatids that we can see, or sister chromatids, which are joined together through centromere. So, the two chromatids, also called sister chromatids, are attached to each other by means of a narrow, very narrow point called centromere. If you can see from this picture that the centromere is there, which is connecting the two sister chromatids. In the late prophase, so prophase again we are dividing into early prophase, middle prophase and the late prophase. Now, we are talking about what happens in the late prophase. So, in the late prophase, fine fibers start appearing around the nucleolus degenerate completely, and by this time the two esters come to lie in the area of future spindle poles. So, I will show you in a diagram that the two spindle poles begin to get connected by fine fibers, microtubules, and the spindle poles are organized without ester in the plant cell. So, see, there is a differentiation point where we can see that how animal cell M phase differs from plant cell M phase. In plant cell division, there are spindle poles generated, but that without the esters, which can be seen in animal cells very clearly. So, these are the three prophase substages, early prophase, middle prophase and late prophase, where we can see in the in the first one the chromosomes have actually started moving to the periphery and the esters start developing, and in the middle prophase the nucleolus started disappearing and the nuclear membrane also started disappearing, and the condensation of the chromosomes is happening, and in the late prophase they have almost moved to the poles of the cells with the ester formation. They have actually, the esters have been formed at the poles of the cell and the chromosomes have started moving towards them and the condensation has been happened. Now we move to the pro-meta phase. Pro means before, meta means second and phases is the stage. So it is before the second stage and after the first stage. So this is a pro-meta phase in which the nuclear envelope degenerates and the differentiation between cytoplasm and nucleoplasm has disappeared, because nuclear envelope is one thing which is differentiating the nucleoplasm from the cytoplasm. That is the differentiation factor into these two things. But now as the nuclear envelope has degenerated, so the differentiation between cytoplasm and the nucleoplasm has disappeared. Endoplasmic reticulum and Golgi complex has disorganized and the spindle apparatus is fully organized now. Spindle apparatus, the mitotic apparatus, we also call it as, consists of numerous fine fibers. Each fiber is made up of 4 to 20 microtubules. So the spindle apparatus will take away the chromosomes to the poles, and going to the different poles means they are developing two different daughter cells, one at one pole and another at another pole of the cell. The spindle fibers converge towards the two ends called poles, and in animal cells the poles are formed by esters, and since there are two esters, the spindle of animal cell is called amphiester. Amphi means both, ester means star. So at the two different ends we have two esters. So that is why in animal cell it is called amphiester cell. The spindle in plant cells is called anestral because there is no esters in the plant cells, as we have already read about it, that in the plant cells the division happens without ester formation. So anestral spindle is also called acentric, while amphiester is also called centric spindle. So these are the terms that has been given to the different spindles. Anestral spindle, acentric, while amphiester, which is present in animal cells, it is called centric spindle. So the spindle apparatus has the maximum diameter in the middle. The area is called equator. If you can see in this late prophase, if you can see in the middle, it is the maximum diameter we can see, and at the poles the diameter is decreasing. So this middle line is called, where the chromosomes are still lying in the late prophase, it is called the equator. So pro-metaphase centrosome cycle. So this is the cycle which I have shown, that the centrosome, the centrioles are dividing, are duplicating, and then they are dividing into two, and then in the pro-metaphase they are actually moving towards the poles and creating those spindles through microtubules. So the fibers of the spindle have negative end near the pole and positive end near the middle. So some fibers overlap in the middle, get connected by bipolar molecular motors and form apparent continuous fibers, others are discontinuous. Which are in middle, you can see them that they appear to be connected to each other, so that is why they are called continuous fibers, while the rest at the corners they are discontinuous, they are not connected to each other. So what actually happening in pro-metaphase, and what are the constituents of these spindles? So chemically the spindle consists of 90 to 95% proteins. Initially also I told you that we will discuss about what are the proteins that are involved in the cell division. So, spindle is again a component of protein with 90 to 95% proteins, mostly tubulin rich in sulfur containing amino acids with traces of actin and myosin, with 3.5 to 5% of RNA and traces of lipids and other substances. So, these are the main components of the spindle. And with the dissolution of nuclear envelope, the central part of the cell comes to have clear fluid areas and chromosomes move freely in this area. Because now there is no envelope which is separating the cytoplasm with nucleoplasm. So, everything is clear now. So, the chromosomes move freely in this area and they enter into the metaphasic. So, now in metaphase, meta is after or second and phase is a stage. So, we are now entering the second phase of M phase, cell division. Discontinuous fibers coming from two spindle poles get connected to the two centromere surfaces or kinetochores of each chromosome. So, now we have seen that there are some continuous fibers and some are the discontinuous fibers. So, discontinuous fibers coming from the two spindle poles get connected to the two centromere surface or kinetochores of each chromosome. Kinetochore fibers or chromosome fibers are also called as tractile fibers. So, these are called kinetochore fibers which get connected to the kinetochores or centriome surface of each chromosome. The central point where chromatins are joined. So, kinetochores actually join kinetochore fibers join to that particular kinetochore and then they lead to the movement of chromosomes. They guide the movement of the chromosome to the different poles of the cell. Each chromosome is attached to both the spindle poles by distinct chromosome fiber, one for each chromatid, and chromosome fibers now tighten. With time in the metaphase, the chromosome fibers now tightens up. The centromere of all the chromosomes form an apparent plate called metaphasic or equatorial plate. Actually, they are present in the form of a circle, and metaphase is the best time to count the number and study the morphology of the chromosome, because now they have contracted to the maximum. Now they have attached to particular kinetochore fibers. So, they form an apparent plate which is metastatic or equatorial plate, right. So, we can count their number very distinctly and read their morphology. So, metaphase is the one phase where most of the studies are conducted to read the morphology of chromosomes and count the number of the chromosomes. So, here we can see what metaphase is. So, there is an equatorial plate that has been formed. It is an apparent plate, it is actually nothing but an apparent plate that you can see with the presence of the chromosome at this particular site or center of the cell and the spindles or esters at the end of the cells or two poles at the end of the cells. So, you can see that discontinuous fibers have connected to the kinetochores of the chromosomes and the continuous fibers have made a straight line in the center if you can see it. So, once this stage has done, that discontinuous fibers have attached to the kinetochores of the chromosomes, the cell enters into another phase which is called the anaphase. Anaphase is up phases is stage. So, it is the up stage of cell division and the centromere of each chromosome divides into two, so that each chromatid comes to have its own centromere now. So, this is the time when the chromosomes divides into two and each chromatid have its own centromere. So, the two chromatids now start repelling each other and separate completely to become daughter chromosomes. This is the stage when we have two sets of chromosomes that are visible. The daughter or new chromosomes move towards the pole of spindle along the path of chromosome fibers or tactile fibers. Now, the tactile fibers or the chromosome fibers are pushing them apart, the two sets of chromosomes. They are already repelling each other and the spindles are also pushing them, helping them to be pushed apart at the two different poles of the cell. They remain connected to each other by interzonal fibers. They still are yet connected to each other. And in anaphasic movement of chromosome, the centromere lead the path while the limbs trail behind, because the centromeres are the one part that are connected to the spindle fibers. So, these fibers are pushing the centromeres up and the chromosomal limbs are led behind or they are trailed behind. That is why different kind of shapes can be seen for chromosomes, that is V shape, L shape, J shape, I shape, depending on the type of chromosomes. Here you can see a V shape where the spindle fibers are connecting or pushing them apart, but still connected to the centromere, and the two sets of chromosomes are still connected to interzonal fibers. They have not separated, but they are pushing apart, they are repelling each other, and the spindle fibers are also pushing them apart, but still they are connected to interzonal fiber and they are moving towards the ends or towards the poles. Or at the end of anaphase, two groups of chromosomes are formed, one at each pole of the spindle. As you can see from this picture, they have moved a wide area, and most spindle fibers disappear from near the poles, but had remained intact near the middle. They are still at the middle, but they have disappeared near the poles, and the number and type of chromosomes at each pole is same as present in the parent nucleus. So, now at this stage the number of chromosome in this particular stage of cell division is double, and the number of chromosome at one particular pole is exactly the same what is there in the parent nucleus. You have to remember this, and these are still connected to each other through different fibers, but the number of chromosomes at each pole is same as that of parent nucleus, but overall the number of chromosomes in the cell have been doubled. So, now we are entering the last phase of mitotic phase, which is telophase. Telos means end, phase means stage. So, it is the last stage of mitotic phase. So, the stage is reverse of prophase. What has happened in the prophase will be completely reversed in this telophase. Cytoplasmic viscosity, we studied about it, that it had increased in prophase, but now it will start decreasing. The two chromosome groups reorganize themselves into a nuclei. The nuclei started disappearing in prophase, but now it will start appearing in telophase, and the two chromosomes will start organizing themselves into the nuclei. Chromosomes elongate and overlap one another to form chromatin. The reverse had happened in the prophase, that chromatin started separating and then chromosomes were condensed in that stage of cell division, but now the chromosomes started elongated and overlapping one another. Nucleoplasm collects in the area of chromatin. Now, nucleoplasm, as we had seen in prophase, that there was no differentiation in nucleoplasm and cytoplasm because of the absence of nuclear envelope, but now what has happened, that nucleoplasm now started collecting in the area of chromatin, because now the nuclear envelope will start forming itself. A nuclear envelope appears on its outside from pieces of older nuclear envelope and annulated endoplasmic reticulum. In the prophase, endoplasmic reticulum and Golgi apparatus have vanished, or they were not there, and now from those parts, those degraded parts from the nuclear envelope or the annulated endoplasmic reticulum, they start collecting each other, and a new nuclear envelope will start forming at the two ends or two poles of the cell to form a complete nuclear envelope or nuclei, and the two daughter nuclei are formed at the poles of the spindle. So, here you can see that in early telophase, cytoplasmic constriction also started happening, astral rays started disappearing, spindle started disappearing, and nuclear membrane started appearing, and nucleolus also started reappearing. This is exactly the same opposite what had happened in the prophase, where nucleolus started disappearing and nuclear membrane also started disappearing. So, telophase, spindle fibers disappear around the poles, Golgi complex and endoplasmic reticulum are reformed, because now the all the factory information has to be restarted in the newly formed cells. So, all these apparatus are required. So, Golgi complex and endoplasmic reticulum are reformed, and then astral rays are withdrawn in animal cells, because there are no astral ray esters in plant cells. So, that is why it has been specified that astral rays are withdrawn in animal cells. So, we can see that they started disappearing. So, at this stage we have understood what and how the mitosis happens in animal cells. So, what are the different phases? We had the prophase, we had the pro-metaphase, we had the metaphase, then anaphase, and then telophase. So, these are the different phases where spindle formation, ester formation takes place. Nucleolus disappears first, nucleolar membrane disappears, and then at the by the time we reach the telophase, nucleolus start reappearing, astral rays start disappearing, and nuclear membranes start forming, with the rest of the organelles organizing themselves as particular daughter cells. So, at different stages we have observed that how the number of chromosomes and at what stage the number of chromosomes actually we can see in a diploid format, where from 2C to 4C, and in the metaphase we can actually read the morphology of the chromosomes, the actual number of the chromosomes. It is the best phase to study any kind of chromosomal changes into the M phase, because this is the phase where the chromosomes are most condensed and have arranged themselves so that it can be read easily. So, different when they move, when the chromosomes move during the cell cycle, it is mainly targeted through centromeres, which are connected to kinetochore fibers that guide them that they need to be moved towards the poles to the esters with the centrioles. So, these are the things what we have, these are the process steps that happen during the mitotic phase. We started from, and once the cell has completed its cell division, till now we have studied about the karyokinesis, that is the nuclear divisions. We have not yet read about the cytokinosis, that is the cytoplasm or complete cell division. We, in the upcoming sessions, we will be discussing about the complete cytoplasmic division as well, how it happens and what is the difference between the cell division of animal cells and the plant cells. We will be discussing that how it happens, what are the factors that affect them, and what is the differences between the cell division of plant cells and animal cells. We will be discussing all those aspects in detail before we move to meiosis, which would be another process of cell division that happens in the reproductive cells. We will try to understand how the different cells proceed, how and why different cells proceed to different type of division, what are the reasons behind them, and what is the outcome of the different divisions that happen with these two different processes. So, we will discuss all these things into the next series of this discussion. Thank you.